When I first heard about the Symposium for Clark, I genuinely wanted to be there in person. Unfortunately, as an Orthodox Jew and a Sabbath observer, I could not travel today. I first met Clark in the early 1980s. I had applied to the MIT ChemE PhD program while I was still a medical student at Yale. Here I was, a somewhat happy go lucky medical student who already had a PhD in immunology, trying to convince the admissions committee that this bizarre trajectory somehow made sense. And then there was Clark Colton, admissions officer, scientist, engineer, and ultimate gatekeeper, quietly trying to determine whether this truly unconventional character should be allowed anywhere near the department. At that point, Clark knew absolutely everything about me: my previous life, my grades, my scores, my background, my recommendations, probably even my psychiatric profile. Meanwhile, I knew absolutely nothing about him. But over time, during that application year, I learned of some remarkable work he was involved with together with Dr. David Terman at Baylor College of Medicine. Dave Terman had discovered something astonishing. He would subject animals and patients with breast cancer to plasmapheresis and then pass their plasma over protein A immunoadsorbent columns and then back into the patients. First in dogs with breast cancer, later in human patients, tumors reportedly became inflamed, became hot to the touch, and in some cases regressed dramatically. Now, to a young physician scientist in training, this sounded absolutely wonderful. This was before immunotherapy became fashionable, before checkpoint inhibitors, before CAR T cells, before every biotech company on earth added the word immune to its PowerPoint presentation. The notion that you might manipulate the immune system through extracorporeal means seemed very exciting. And Clark was right in the middle of it. What struck me was not just the technical sophistication required, it was Clark's intellectual style. Clark had the ability to move fluidly between engineering rigor and biological imagination. He could think mechanistically, mathematically, and biologically all at the same time. And for many of us, that became a model of what biomedical engineering should aspire to be. The Terman phenomenon immediately triggered mechanistic ideas in my head. I became convinced that at least part of what Dave Terman was observing might involve the manipulation of circulating immune complexes containing tumor antigens and antibodies directed against those antigens. The hypothesis we developed was that these complexes might normally exist in forms that were immunologically ineffective, essentially sequestered or functionally inert. But if protein A immunoadsorbents could alter the structure, composition, and binding dynamics of these antibody containing complexes, the procedure might liberate or reconfigure components capable of mounting a meaningful anti-tumor response. In other words, the adsorption process might not simply be removing factors from the circulation, it would actually be reshaping immune interactions. We speculated that this could trigger downstream immune activation against tumors through altered immune complex processing, FC macrophage interactions, and complement activation, and broader modulation of the host immune response. So, like many young scientists blessed with excessive confidence and insufficient adult supervision, I immediately concluded that I had the solution. I wrote the entire concept up as an NIH R01 grant and promptly shoved it into Clark's hands with instructions to submit it to the NIH. Now, was often the case with written products emerging from Clark's students and postdocs. Clark wanted to wait for the next submission cycle. He wanted to think things through more carefully, refine the proposal, etc. At which point I essentially informed him that if he did not submit the grant immediately, I would not be coming to MIT. So he submitted the grant, and to our mutual astonishment, it was funded, which of course immediately convinced me that I was a scientific genius, a belief that survived for a few weeks before reality re entered the conversation. The following year, I arrived at MIT, began working on the project, and this is where the story becomes very MIT like. What began as an attempt to explain a clinically observed cancer phenomenon evolved into a deeply fundamental basic science project focused on the structure, size, and composition of monoclonal antibody-based model immune complexes using quasi-elastic light scattering and rotary shadowing transmission electron microscopy. Regina Murphy, who was a graduate student at the time and who went on to have a stellar career at the University of Wisconsin ChemE department, joined the effort, and together we worked on trying to understand these antigen antibody systems at a much deeper biochemical and biophysical level. But here's the important part. We never solved the original Terman problem. And that turned out to be okay because over time, the excitement surrounding protein A immunoadsorption gradually faded. Not because it was fraudulent or the observations were wrong, but because the underlying biology turned out to be much more complicated than anyone appreciated. The protein A columns were biologically messy systems. They broadly removed immunoglobulins, altered immune complex distributions, perturbed complement pathways, and affected multiple layers of inflammatory signaling simultaneously. Also, clinical reproducibility became problematic. Some patients appeared to respond dramatically, while others showed little benefit. Tumor regressions were inconsistent and difficult to predict. Large controlled trials failed to produce the kind of overwhelming signal needed to establish the technique as a part of oncology practice. As more patients were treated, the effect appeared less spectacular than the original reports suggested. Finally, the field of oncology itself changed rapidly underneath the technology. During the 1980s and 1990s, several revolutions occurred almost simultaneously. Monoclonal antibody technology matured, recombinant cytokines emerged, molecular oncology exploded, oncogene biology advanced, targeted chemotherapy improved, and eventually checkpoint blockade and CAR T cell therapy appeared. Compared to these newer approaches, protein A immunoadsorption began to look technologically cumbersome, too expensive, poorly mechanistically defined, and difficult to standardize. But in retrospect, I believe something genuinely important happened there. The field was reaching towards concepts that modern cancer immunotherapy would later rediscover in far more sophisticated molecular form. The intuition that tumors can create systemic and local immunosuppressive environments, that circulating factors matter, that immune complexes might shape immune behavior, those ideas were not crazy at all. Clark and I continued working together over the following years on a series of highly interdisciplinary projects, ranging from developing large-scale immunoadsorption system to early efforts in the development of the bioartificial pancreas and bioartificial liver. It was an extraordinary intellectual environment where engineering, biotechnology, biochemistry, cell biology, immunology, and medicine constantly intersected. Probably the most enduring lesson I learned from Clark was the meaning of true scientific rigor and the importance of robust quantitative and statistical analysis. Coming from the Rockefeller University, I had already been immersed in a culture of creativity, big ideas, intellectual risk taking, and thinking far outside conventional boundaries. But much of my work up to that point had been largely qualitative and intuition driven. What struck me almost immediately about Clark was the discipline with which he approached data. I still remember looking at the graphs he produced, plots containing what seemed like an absurd number of carefully collected data points, all assembled with almost obsessive precision. And most importantly, beneath the engineering and mathematics was a profound intellectual humility. The conviction that the job of a scientist was not to force reality into a preconceived narrative, but to remove the noise, bias, and self deception so that reality could reveal itself on its own terms. During those years, I spent many evenings in Clark's office, smoking cigarettes and chatting about all sorts of bizarre and fascinating subjects completely unrelated to the science at hand. At that time, the world was rapidly converting from paper to digital information, and Clark used to worry that someday somebody would simply pull the plug on all the computers and civilization would suddenly find itself back in the ancient world trying to reconstruct knowledge from memory. Another time, we tried to estimate how many atoms existed in the entire universe. And we speculated that if we actually knew the number and had enough computational power to simulate the system with molecular dynamics or Monte Carlo methods, we could essentially become God. And then, of course, there was our occasional campaign against the engineering nerds. Clark and I would sit around inventing qualifying exam questions specifically designed to rattle their cages, disrupt reflexive thinking, and remind students that intelligence was supposed to involve awareness of the outside world beyond equations and transport coefficients. One question I remember asked simply, what is air supply? The engineering students would confidently write that it was a company supplying industrial gases or ventilation systems. We would then, of course, gleefully mark them wrong. The correct answer, obviously, was the Australian soft rock group Air Supply, that was very popular at that time. That was part of the joy of being around Clark. He was intellectual without ever becoming pompous. He was grounded, direct, sometimes mischievous, and refreshingly honest about people and ideas. That fit perfectly with my own no bullshit personality. You always knew exactly where you stood with Clark. And of course, there was another characteristic of Clark that many people here, I'm sure, remember. He had the remarkable ability to let manuscripts sit on his desk for months and occasionally years, to the growing frustrations of generations of PhD students and postdocs. Oddly enough, that never bothered me. Coming from my background, I already knew how to write and push papers forward. So if Clark disappeared into one of his long manuscript hibernations, I would simply keep moving and publish the work anyway. But one of Clark's lasting contributions was deeper than any single paper or project. He helped shape a generation of people who believed engineering principles could fundamentally transform medicine. Today, everybody says bioengineering as if the field always existed in its current form. It did not. People like Clark built that intellectual bridge before it was fashionable, and before universities started putting bio in front of their engineering department names. Clark helped create the mindset that biology was not too complicated for quantitative analysis, that medicine could benefit from engineering rigor, and that difficult problems were invitations rather than obstacles. People here today were influenced by him in different ways, some through direct mentorship, some through collaboration, and some simply through proximity. For me, Clark represented something increasingly rare. He represented intellectual honesty. If something did not make sense, he did not pretend it did. If an idea was weak, he was not going to decorate it with buzzwords, hype, or fashionable optimism like many of the now famous bozos at MIT and Harvard. He respected reality too much for that. And frankly, in today's scientific culture, that quality is becoming increasingly precious. So, although I could not be there physically today, I wanted to take a moment to simply say thank you. Thank you, Clark, for your mentorship, for your rigor, for your seriousness of purpose, for your intellectual honesty, your curiosity, your humor, and your refusal to tolerate sloppy thinking. And most importantly, thank you for helping shape generations of scientists and engineers who continue to carry your influence forward every single day, often without even realizing how much of their scientific DNA came from you. The older I get, the more I understand that truly important mentors in life do something far greater than teach techniques, supervise projects, or help publish papers. They fundamentally alter the way you see the world. They permanently elevate your standards for how to think, how to question assumptions, how to analyze evidence, and how to pursue truth with discipline and humility. Clark did exactly that. His fingerprints remain all over modern bioengineering and modern medicine, whether people recognize it explicitly or not. Thank you all very much for your attention, for your participation, and for helping honor a truly remarkable scientist, mentor, and human being. Good Shabbos, everybody.
Well, I don't see my video image, but my name is Robert Hillman, and I want to greet you all from California. And although Clark and I haven't been in touch a long time, I wanted to make an observation that I think after hearing you, we'll see is consistent with all you've been saying. So back in 1976, fresh out of a Stanford chemical engineering, bioengineering undergraduate, and work at a pioneering company called ALSA, who was pioneering in drug delivery systems at the time, I came to MIT and wanted to work on closed-loop drug delivery systems. Clark at the time had a collaboration with Stuart Soldner at the Joslin Clinic on developing an implantable electrochemical sensor with a company called Giener Inc. And at Joslin, there was a bright physician named John Guyton Jr., who had developed and programmed a model of glucose and insulin metabolism in a language developed by Jay Forrester, who some of you may remember from MIT, who developed core memory at the Sloan School. It was a visual, symbolic language that could simulate differential equations for non-mathematicians. Well, here's my story. I proposed to Clark that we take that model and look at various scenarios that involve how a glucose sensor and infusion pump would be placed in the body, the location, the response time, and all the dynamics involved with using such a system in a patient. And I'll never forget the first thing he said to me, again, which is so consistent with what many of you said, was that was fine, but I had to translate that model into true differential equations, run it on our computers in MIT, and I never want to see the output from Dynamo. Just translate things into rigorous engineering terms, which we set off to do quite successfully, including some exciting experiments on diabetics at the clinic, and not rely on these soft models and software. When I came to write my thesis, again, another hallmark of what you've all said about Clark is I wrote the thesis, so I talked to him about how to put it together, and he said, give me too much material. Give me something to work with. Give me something to sculpt rather than too little. So that really set the tone for my career. I went on to the ETH Zurich and worked with another MIT graduate, Dick Buckles, who was also a student of Alan Michaels, and worked on drug transport in the subcutaneous space where we did radiotracer experiments. And then I had the unique fortune to work on a closed-loop drug delivery system. It was in the cardiovascular system, which was the first drug delivery system approved by the FDA. I'm now running a biotech company. We've just finished phase 3 clinical trials and have novel clearance for a drug to be given subcutaneously again that can preserve blood flow to the heart in the presence of a blockage. So what I really wanted to say is from those humble beginnings, walking into Clark's office led to a fascinating career at the interface of medicine and engineering and a rigorous approach to mathematical modeling to help understand all of that. Thank you.
My talk is inspired by the classic work by Dr. Seuss, Oh, the Places You'll Go. But it's also a story about how MIT and a PhD in the Colton lab really changed the course of my life. This is where I was born, in the backseat of a car speeding to the hospital. It shows how independent my mom was. We turned around and went home afterwards. When I was 10, we moved from California to a small farm in Idaho. Our house wasn't exactly this, but it gives you an idea of what it looked like. By the time I finished junior high, my stepfather had left, and I inherited taking over our 80-acre farm. During the summers, I spent stacking hay bales, as well as trying to turn what should have been a desert into irrigated farmland. In the fall, I ran a John Deere 95 combine, thrashing grain and other crops. Winter in Idaho was particularly challenging for raising livestock. The temperatures were freezing cold. Often when things would thaw, they would get wet, and animals would get sick. We raised baby lambs, we raised baby calves, along with everything else. When the lambs got sick, we brought them into the kitchen. When the baby calves got sick, we brought them into the kitchen. My bedroom was in the basement. If they got well enough to get out of their cardboard boxes, I'd hear their feet tapping around linoleum floors above my head. This wasn't our school, but it gives an idea of sort of what it looked like going from California to Idaho. The first year, they passed out the math books, which is the same math book I'd used the year before in California. 2 weeks later, they said, turn the books back in. They're clearly too hard. This was me the night before taking the ACT exams. I was baling hay until 3 o'clock in the morning. You bale hay at night because the dew keeps the leaves from falling off the stalks as you're baling the hay process. You may get the idea that living and running a farm was challenging. It certainly was. But I also have to say it was probably the best preparation for practical engineering, both biology in terms of financing, that I could have possibly had. You're keeping livestock alive, you're growing crops, you're trying to keep old equipment running. I left the farm and went to a year at the University of Idaho, where I stayed in farmhouse fraternity. I then served a mission for 2 years for our church in Barcelona, Spain. Por eso hablo español. I then finished my undergraduate degree at Brigham Young University. When I was talking to my advisor, Dr. Ken Solon, about what I should do next, he said, there's this guy at MIT that I think you might want to meet. That brought me here with the original goal in terms of my thesis was to develop a bioartificial pancreas, a rather auspicious goal. The thesis became, however, measuring the effect of hypoxia on insulin secretion and viability of isolated islets of Langerhans. To give you an idea of what we're talking about, the figure on the left is an islet of Langerhans, 1,000 to 2,000 cells, showing the vascularization system, and just how vascularized those islets were is shown by a slide produced by Susie Bonner-Weir, in our audience today, of a polymer cast of the capillaries inside of the islet, suggesting the possibility that this tissue needed a lot of oxygen and transfer capabilities. Our goal was to take and isolate these islets of Langerhans, enclose them in a semi-permeable membrane to be able to translate into people with diabetes and have ongoing insulin response to their blood glucose levels. What we discovered very quickly was that observation of the vascularization was there for a reason, and that is insulin secretion was acutely responsive to decreases in the oxygen levels, responding within 30 seconds once oxygen levels were lowered. Over a longer term period of time, we saw that islet cores develop hypoxic or dead cells in the middle of all the islets due to the lack of oxygen penetrating and their high oxygen requirements. This work led me to my first job in industry, a company called Cytotherapeutics, founded on the same kind of principles that we were trying to develop in the lab. That was immunoisolated transplantation, where we were encapsulating islets of Langerhans to treat diabetes, adrenal cells to treat pain, and chromaffin cells to treat Parkinson's disease. We made multiple different kinds of membranes, including hollow fibers, microcapsules, and flat sheet membranes to be able to determine and enhance the oxygen levels inside of to support the inside tissue. However, no matter what we did, the technology failed. And it failed due to our old nemesis of hypoxia overcoming the capability of the islets to stay alive and respond, or the tissue to stay alive and respond to the outside stimuli. What I really learned at Cytotherapeutics, though, was a startup culture. It was, excuse me, work hard, play hard, and we did both. Learned about science and financing as we faced partnerships with other large pharma companies, about teamwork and working together across disciplines, about business development in terms of trying to induce others to get excited about what we were doing, and maybe most important, about the impact of leadership. I was fortunate to have Mark Levin, one of the pioneers of the biotech era, be our CEO and really guide us and inspire us despite the challenges that came up along the way. When Cytotherapeutics changed its direction because of the challenges, I moved back to California. Took a job at Alza, which was infamous at the time for its osmotic delivery technology. They made the Alzette, which was a 3 to 14 day osmotically driven drug delivery system that many researchers use to deliver drugs to their animal models. They also had a second program called VITS, Veterinary Implantable System, which was used to deliver growth hormone for 14 days to pigs and cows prior to slaughter. It consisted of a sleeve membrane on an osmotic engine, a salt tablet, a barrel containing the drug, and then a narrow orifice for releasing the drug. When I arrived, my job was to develop a human implantable therapeutic system, the goal being 360-day delivery of leuprolide to control people with prostate cancer. Originally, it was envisioned to look very much like VITS, a slightly larger model. However, when I got there, I decided a few changes were needed, each one facing different levels of resistance from management. To begin with, I replaced the sleeve membrane with a plug membrane to deal with the lower flows that were required for the long-term system. On the diffusion moderator, replaced the narrow orifice with a screw, literally, that created a long, narrow orifice to prevent ingress of fluids from the outside in the body back into our drug formulation. However, what created the most resistance by management was the proposal to switch from cheap injection-molded polystyrene bodies to titanium bodies. The first cost about a penny a piece, the latter cost $15 a piece. Despite all my efforts, management refused to spend the extra $15. Out of desperation, I invited the CEO, Ernie Mario, Alejandro Zaffaroni, the founder of Alza, as well as the rest of senior management, to a demonstration. I went and bought a series of chicken breasts, filled the devices with red dye, and invited them to come and witness the effect of hitting them with about a 40-mile-per-hour baseball. Ernie and Alejandro were picking up pieces of chicken breast off their suits and red dye that covered them, but 10 minutes later I received permission to move to drilled titanium to develop the system. Ultimately, we were successful. We developed a system that delivered leuprolide acetate at a zero-order level for 360 days that was approved for treating patients with prostate cancer. At this point in time, I moved on from Alza because I got a call from Mark Levin, my previous CEO, who was at the forefront of the molecular biology revolution that was just hitting, the potential to sequence the human genome and understand biology at a molecular level. Mark approached me and said, "Keith, I'm looking for engineers who speak biologies." My response to him was, "Mark, I don't even know how to spell DNA." Because he trusted me, he hired me anyway. The first thing I did was buy a molecular biology textbook and started on page 1. And since I was commuting from California to Boston at the time, my wife was pregnant, I was able to finish the book and learn at least a little bit about what I was going to be doing. Things moved fast at Millennium Pharmaceuticals. It was one of the 3 big drivers in terms of the molecular biology era. I started off as the program manager of a collaboration with Astra, $40 million collaboration, where we were providing our molecular biology tools to them to use for discovering targets potentially valid in the area of inflammation. Shortly thereafter, we launched the Millennium Monsanto Agricultural Alliance. I was made the head of that because, having come from a farm, I did know the difference between a potato and a tomato, unlike many of the other collaborators. This was a $200 million agricultural collaboration, again, providing them with our technology, as well as starting up a company next to Millennium that we would co-manage that had the opportunity to earn another $100 million in milestones for successfully meeting its objectives. Shortly thereafter, I was made the president of Millennium Technology Solutions business. It was a $100 million a year business unit where we did multiple partnerships with a number of large pharmaceutical companies, as well as in-licensed new technology from well over 50 different companies that had various capabilities we wanted to integrate into our platform. When the molecular biology bubble burst, Mark asked me to lead the restructuring of Millennium because by then I'd worked across every division within the company. The gutsiest challenge I faced in this whole process was really leaving my knowledge bases. I literally knew nothing about molecular biology when I accepted the job. Having worked now at both in technology and in business development, I received a number of offers to be the CEO of a number of different biotechnology companies. The one I selected was Elantos, a company located both in Heidelberg, Germany and Cambridge, Massachusetts. It was based on Nobel laureate technology, but on my first visit to Heidelberg as the CEO versus prospective CEO, I looked at a lot of data that was being ignored and realized the technology does not work. I came back to the first board meeting and informed the board members we had a choice. We could refocus the company or close the company. I think the members were shocked enough that a first-time CEO in his first board meeting would even suggest closing the company that they said, let's give this kid a little bit of time and see what he can do. We took an awful lot of chances, but during the next 3 years, we put 3 compounds into development. Some of the chances were we declared a development candidate based on efficacy in a dog without ever testing PK. It turned out, in fact, there was no PK. But that led to the discovery that most DPP-4 degradation occurred in the gut as opposed to the systemic situation, so the drug actually worked, which drove acquisition by Amgen for $300 million for the company. The gutsiest challenge I faced was completely shutting down the technology on the Heidelberg site. But I knew if I didn't, because I wanted desperately to have something, but I knew if I didn't shut it down, the Heidelberg scientists would continue to work on it because that's what they were hired to do and that's what they knew. My next job was as a CEO of a company called Surface Logics. My mandate was to restart a company that had just suffered a failed phase 2 clinical trial. The company had 3 programs. We repeated the phase 2 trial and it failed the second time. I killed the second program when he discovered that our molecule was actually covered by a Merck patent. The third program we advanced. It was a selective ROCK2 inhibitor. It had some amazing characteristics. By the time to try to figure out where it would really work, we were hit by the biotech recession in 2011. The best we could do was raise enough money by selling the company to be able to pay our bills and shut down legitimately. But we also added a royalty on any future products. Well, that third program became Rezurock, approved for chronic graft versus host disease, currently sold by Sanofi. Last year's revenues were over $500 million. It was an exciting time, and I could reach back to all of our employees and say, I need an update on your email addresses because you're about to receive royalty checks. After Elantos, I moved to become the CEO of Constellation Pharmaceuticals. Constellation was based on epigenetics. You don't have to ask, I knew nothing about epigenetics. But it had a major collaboration with Genentech. My mandate walking in was to manage the collaboration, select development candidates for 2 leading targets, and then advance them through phase 1, phase 2 clinical trials, which would secure a preformed acquisition by Genentech. Well, a year into it, the public markets opened up and the mandate changed. Negotiate a Genentech exit and then take the company public. However, despite all my efforts, Genentech's consistent response was, no deal. We like what we've got, we're going to acquire you. I heard that message up until the day before the acquisition was to be announced. And then the message was, we changed our minds. We're not going to tell you why, but we're not going to acquire you. Well, the new mandate then became to raise money while explaining to investors why it was Genentech passed when it was well known that they were going to acquire us, and then re-recruit a team, many of whom already had job offers from other companies, and then I departed. Constellation Pharmaceuticals was later acquired by MorphoSys for $1.7 billion based on those two molecules that we discovered, took into the clinical trials, and the Genentech rejected. The gutsiest challenge I faced was dealing with what happened after Genentech said no without telling us why. It wasn't until years later that we realized we had two molecules. Genentech wanted one, Roche wanted the other. They couldn't decide between them how to pay for it and split the cost, so they said they were going to pass. It's amazing how some companies will do things. Following Constellation, I became an entrepreneur in residence at Third Rock, and then became the CEO of ChemE, which I helped launch. ChemE was based on autophagy, the ability of cells to degrade large products inside of themselves to clear them out. But soon thereafter, we were hit with COVID-19 and a number of different questions. How do lab workers work from home remotely? It's pretty challenging to take the hoods home with them. Demands for outrageous promotions months after someone would arrive, for salary increases that were astronomical, as well as for DEI hiring above someone's qualifications. This really led to me being one of the 7 drivers of forming a collaboration of CEOs initially across the Boston area to be able to share information about how to work in a COVID-19 environment and other issues. That's now grown to over 100 CEOs that have weekly meetings to share information to try to help each other be successful in developing drugs to treat human diseases. But it turned out that at ChemE, all 3 founders' programs failed to validate. We overall spent $150 million to try to validate those programs and then discover one new development candidate. The result of that is I was fired. This is biotech. Not everything you do works. The gutsiest challenge I faced was really telling the academics that we were killing their founder programs because they didn't work the way they thought they did. Many of you can understand how if this is your major program, to hear somebody say it really doesn't work the way you think it does, and you've published, we got an awful lot of flack coming back. Currently, I'm the CEO of Luxa Biotechnology. Luxa is developing adult retinal pigment epithelial cells to treat patients with dry age-related macular degeneration. To give you a size comparison, there are 8 employees at Luxa. There are 200 million patients worldwide who suffer from dry age-related macular degeneration, loss of their central vision. However, we've made some major advances. We developed a manufacturing process where we can take eyes originally donated for corneal transplants, extract a retinal pigment epithelial cell stem cell, and expand it to be able to treat up to 3,000 to 5,000 patients from one set of donor eyes. From the FDA, we've received RMAT, or Regenerative Medicine Advanced Therapy, Breakthrough Therapy designation for our program going forward. We're currently dosing the final cohort of our phase 1/2A clinical trial. To give you an idea of some of the potential of this product, these are the results from the first cohort of patients we dosed who were almost legally blind at the time they received their implants. On average, now 360 days after being implanted, they can see 22 letters better than they could before receiving the transplant. 22 letters equates to between 4 and 5 lines better on a diabetic eye chart. Truly a remarkable change if we can maintain this and help others to be able to see the way this initial data is starting to look. What's my gutsiest challenge at Luxa? I don't know yet. We'll still find out, but right now it's a fun ride, and I have an exciting time moving this forward and going back to my early days of cell therapy. Let me go back a little bit and just say what were the biggest lessons I learned from MIT. First of all, it was to learn how to learn. Take chances, survive setbacks, have the scientific humility to know when I was wrong, but also the confidence to know when others are wrong. To be willing to take risks, even in new areas. That's in science, biking through Boston, as well as climbing Mount Kilimanjaro. but also enjoying the ride. MIT wasn't all about academics. We did a lot of other things as well. We played soccer, volleyball, ice hockey, water polo, ran the Boston Marathon. We danced and formed lasting friendships that last until today. It's truly an exciting time. Let me step back and say what happens, what about life outside of work and everything else? Well, it's been exciting. We have 4 active kids. One is an orthopedic surgeon, one is an accountant, one is studying econ, one is an engineering robotics graduate. I was the bishop of our Cambridge church for 5 years with a group of about 100 different students and others coming up through Boston. I served in our church's Boston mission presidency for 3 years during the COVID period. I've run over 10 marathons, completed over 10 triathlons. My kids calculated I biked over 50,000 miles, biking into Cambridge on a regular basis and basically throughout lots of parts of the world, including 2 200-mile one-day races from Logan, Utah to Jackson, Wyoming with my adult daughter. That included 9,000 feet of climb in those races. I've coached all 4 of our kids' soccer teams. I play wing in an over 62 competitive soccer league. We're currently in 2nd place. Have a game tomorrow. My wife, Elizabeth Dion, who many of you met last night, is running for Massachusetts State Treasurer. If you're a Massachusetts resident, vote. I thought about retiring, but not yet. Life is still too exciting. Thank you.
Good afternoon. I am not a student of Clark's. I did not do anything at MIT, but I was one of a pair of a long-term collaboration of my husband, Gordon Weir, and myself with Clark, really looking at the oxygen demands of the islets of Langerhans. And does this make it go? Right arrow, all right. So Gordon unfortunately cannot be with us, but he had a very simple thought for many years that diabetes was really a simple disease, that there are just not enough beta cells to handle the demand for insulin. And there was so what do? Well, you either replace them or you grow them. And his sort of research went off mainly to replace them, and mine was really to grow them. But we worked as a team and shared a lab for about 40 years. We have a true obsession with islets. That's our license plates. You can tell us who's driving in front of you if you see that. But I think that what really sort of made a big difference in an area of our research, particularly what Gordon's wanting to replace the beta cells, was when he was on both Keith's thesis committee and then Stathros's committee. And I remember when Gordon came back from a committee meeting with sort of this diagram saying, Clark's team has figured out that you really, with an isolated islet, the cells don't have enough oxygen in the center, even if you're only 150 microns in diameter, that the ones in the center are not going to have as much oxygen as they're used to having. And so essentially, I guess it was a little bit in between the 2 theses that Gordon said, we got to work together. And so we did work for many years then with 22 publications. Gordon and I were, I think, almost permanent members on his graduate students. I know he had many more graduate students, but between the 2 of us, we were on a graduate student or 2 at every time, all the time. And what we worked on mainly was the issues of packing density, microcapsules, macrocapsules, how could you enclose those islets, as you heard earlier about the encapsulation, while maintaining both the islet function and the survival of the islets? And then, at a later time, once Clark's group started working on human embryonic stem cells to be a source for new islets, we were on committees for both Jeff and Daryl and Amanda too. So all of this, obviously, we had been fairly rigorous, but we were doing it more from a medical doctor or a biologist point of view. But with Clark, we ended up having much more rigor and quantitation, and the formulas we had to sometimes put the formulas, say, no, not for this paper. That can go in the appendix. That's not going into the main text. So, but what I think through the years and many different types of experiments, we basically were able to show that microcapsules here on the left, which were of adult pig islets in little alginate capsules, or more a macro capsule like the Theracite or the Baxter device that we used in the lab. Encapsulation of islets did protect the islets in rodents, but not in bipeds. What I think one of the really interesting studies based off of the interaction with Clark's lab was this paper, which was Gordon's technician of Esther O'Sullivan and Amy Johnson, who was Clark's student, is that if you broke up the islets and made them in smaller aggregates, they survived that much better in microcapsules. And when you then transplanted just sort of a very marginal amount of the islet tissue into a diabetic mouse, and we were using xenographic, so there were rat islets into a diabetic mouse. On the left, if you just, on the left, if you just see what you did, if you put in islets in the capsules, nobody cured in the 2-week period. But if you did these aggregates with only 85 of the equivalents of the islets, that you ended up 10 out of the 12 of them became non-diabetic, recovered their glucose homeostasis. So Esther did this part of the work, but then Amy ended up doing the calculation to show, yes, if you really did the calculations, it's because all the islets now would be able to have proper oxygen for proper function. And as Keith already showed you, using from my microvascular casts, the islets are normally very well vascularized. So this was the sort of thing that we could do back and forth, we providing the islet work and the morphological side of it, and Clark's having the engineering questions about the oxygenation, the transport of oxygen and diffusion of the other substances that were important for the islet's health. Gordon had, for many years, and I think this was the stem of, we shared lab meetings often with Clark's group, that the problems of encapsulation were, how many islets can you pack in the same space and have enough oxygen? What was the kinetics of insulin release? And then even for the microcapsules, is if you transplanted them into the peritoneum, how will they survive? And this became particularly true once you got into either monkeys or theoretically into humans because of being bipedal rather than four-legged. But I think there were lots of ways that could be used to improve microcapsulation. Some of these have been tested often by some of the people in this room. But I think it came down to a few years ago, well, it looks like 10 years ago according to the slide. I took one of Gordon's slides that was from a JDRF had a encapsulation consortium for a number of years. It evolved into a beta cell replacement consortium, but initially it was encapsulation. Gordon had been asked to be the chair of the consortium, and at some point he was sort of giving this talk that the encapsulation issues that kept him awake at night were the perm selectivity, the oxygen requirements, and the insulin release kinetics. And I think he was convinced that the encapsulated islets would not work in humans, and that the recent data from clinical trials, both ViaCyte and Vertex, basically have supported that conclusion. There may be new formulations using some of those possible ways of making macrocapsules better that may end up working, but there's hard challenges that have to be met. So we learned a lot from the Colton lab. Hopefully they learned from us. We had a lot of fun. I warned Daryl that I had a picture of him. This was the only one I had, but it was from the cell transplant meeting. It's the only one I had of them. And there were a number of our group who basically had worked for many years. And I'm sure all of those who worked on the projects that were in conjunction with Clark understood a lot more about the rigor that one needs when one's doing this sort of experiments. So I too want to thank you, Clark, for what you provided us, and obviously from what I've heard, how much you've actually, for the students that you've had. Thank you.
Hi, I'm Bob Johnson, and I'm going to talk about Clark's involvement in a cellular encapsulation project at Baxter Healthcare's gene therapy business unit back in the 1990s. The project started with a focus on diabetes, but evolved into genetically engineered cells. My relationship with Clark is different from most in the room today. I'm not a former student or postdoc. I first met Clark in an elevator at Baxter Healthcare, I think it was 1990. He walked in with another Baxter employee and looked around at all the people inside and said, "I'm looking for some guy from Princeton who's working on diabetes." That was me. At the time, I was research director of a new project we called immunoisolation, and was part of a broader initiative to evaluate the feasibility of an artificial pancreas. Clark started working with us as a consultant. However, his involvement rapidly changed to become a project participant and collaborator. The data reviews with Clark involved a great deal of time at the microscope looking at the latest histology. There was always a lot of excitement when Clark visited because we knew that there would be a bunch of lively conversations and at times one, maybe even animated discussions as we looked at the latest data, and he encouraged the entire team. However, before we began reviewing data, we wanted to hear a critical piece of information from Clark. Where had he abandoned his car at Logan Airport? We knew he would probably be in a rush to catch his flight to Chicago and he wouldn't have time to park it. We liked his stories, but we worried that he might be arrested on his return. So what I'm going to do is show some slides that are snapshots of things that Clark worked on when he was working with us at Baxter. So back in the 1980s, late 80s and early 1990s, there was a lot of interest in trying to transplant encapsulated porcine islets to treat type 1 diabetes. However, the literature was filled with reports of implanted devices that failed due to fibrotic encapsulation. So we created two teams at Baxter to address the problems. One is to look at the encapsulation issue and see if we could prevent the fibrotic capsule. And the second was to look at the immunology of trying to transplant porcine islets. This is a picture of the people that were in the group that Clark worked with. And they stuck me in at the top because I was going that day, I guess. But below me, there's an arrow pointing to the person that Clark with very intensely. That's Jim Browker. And Jim Browker was in charge of the tissue in reaction to a membrane project. I hired Jim as the first employee of the immunoisolation team, and it was done at a FACET meeting on May 4th, 1988. And the reason I remember that date is because there was a huge explosion of a rocket fuel plant in Henderson, Nevada, which was about 12 miles away, and the concussion blew in the doors of the exhibit hall. Jim did a great job in the screening of membranes. He later went on to become the first VP of R&D at
Bob,
Dexcom.
quick interruption. Just so you know,
Yeah.
your slide is still stuck on your title slide. If you can move that down to what you're talking about.
Geez, I'm not sure why that's the case. Let me see. How about now?
Still the same.
Okay.
You're saying you need to go into presentation mode.
It was in presentation mode, but let's do that again.
Yeah, that one.
Okay. Do you see the second slide now?
No, nothing's changed. Can you click that icon that you're mousing over right there? All right, at least we're on the second slide.
Is it on the second slide now?
Yes, but it's not in presentation mode.
Sorry.
There
How
we
about
go.
now? Let me see if it change. Does it change slide now?
Good to go. We'll leave it. You have the floor.
Okay, good. Well, I was saying that this is a picture of the team that Clark worked with, and I had an arrow showing Jim Browker underneath my picture. And Clark worked very closely with Jim, and Jim went on to become the first VP of R&D at Dexcom. And while he was at Dexcom, he also used Clark as a consultant. So when Clark visited, this is the kind of thing he often looked at. I hope you can see the histology now. On the left, there is a tight pore membrane with a fibrotic capsule forming around it, labeled with the F. But when we screened membranes, and we actually screened 150, we found a pattern emerged, and that was that when cells could enter the membrane, we often got vascular structures forming at the interface. And this was independent of polymer chemistry, but we stuck with PTFE polymers because we knew that the FDA was very familiar with their use in medical devices. So that large membrane that allows cells to get into the pores obviously isn't going to protect anything. So we used a smaller pore membrane that had 0.45 micron pores. And this is a very large pore, actually, compared to what a lot of other people were looking at. We wanted to have a membrane that was as wide open as possible to allow diffusion of macromolecules across the membrane, but also tight enough to prevent cells of the host immune system from getting across. And that kind of laminate was used to create a therapsite, which you're seeing right here in cross section. And there's a center opening for cells, and then you can see different membranes as you go away from the center core. The outside is a woven polyester mesh for structural integrity. Now, if one puts islets in this kind of device and then puts it into a fresh site prepared in an animal, I would expect most or all of the islets to die. But if it's implanted first and allowed to pre-vascularize and heal in place, then you have much better success. In fact, our colleague Annika Tabel at the Karolinska reported that She can use 1/10 the number of islets to cure her diabetic mice, which she allows the device to be pre-implanted and vascularized for 3 months. So we thought that we had a combination that would prevent the fibrotic encapsulation, but what about the immunology? And we looked at allografts and xenografts, and the bottom line is that we could protect allografts, but we could not protect xenografts. And here's an example of an allograft experiment. Over on the left, there's Sprague-Dawley fetal rat tissue, lung tissue into Lewis rats, and in an intact device, they form vacuoles with ciliated cells. But if you poke a hole in the membrane, you get an invasion of cells from the host's immune system and destruction of the tissue that's inside. The situation was much worse for xenografts because whenever we put xenogenic tissue into an intact device, we saw a huge inflammation occur around the device and then death of the tissue inside. In this case, it's mouse fetal lung tissue into Lewis rats. So this told us that the initial idea, the feasibility of implanting pig islets into humans was probably not going to work. And the diabetes project was shifted away from Baxter to a company on the West Coast called Neocrin. And at Baxter, we continued on with looking at allogeneic cells inside our devices. And here's an example of a human cell line, a fibroblast cell line called MSU 1.2, that we genetically engineered to express human growth hormone. It was implanted in nude rats. And when you first do an implant with a filled device, it's common to see cell death in the beginning, naturally because of the ischemic period. But then with these cells, they would recover and start growing inside. And you can see the rise in HGH until the point where we explanted the device and it fell back down to baseline. We did allograft experiments in many different species, including non-human primates, along with our collaborators, and they all showed that the device would protect allografts, but we knew it wouldn't protect xenografts. Our colleagues at the Karolinska wanted to move forward quickly and take a look at the ability of the devices to protect allogeneic tissue in non-immunosuppressed human volunteers. And so Annika Tabel loaded devices with human parathyroid tissue and implanted them in patients for up to a year. And what you see here is a volunteer's arm with 3 of our large devices that contain 40 microliters of volume implanted in the patient. And on the right, you can see the scar where the incision was made for the blunt dissection. Now, Annika put in some small devices that were used as sentinels to see how the parathyroid tissue would do. And she implanted those for 4 weeks. And then when she explanted them, you can see some histology on the left where there are parathyroid cells, but also a lot of fibroblasts in the mixture. She knew that it was an impure preparation, so that wasn't surprising. And on the right, there's a histology after a 12-month explant, and you see some parathyroid cells, but you see a lot of fibroblasts that grew out. But nevertheless, this showed us that, in fact, these devices would protect allogeneic tissue when implanted into non-immunosuppressed humans. So the thing that we really wanted at this point was a beta cell line or an engineered beta cell that we could grow out and place in these devices. We were skeptical about islets because of the oxygen and nutrient demands as have already been described. So this is a picture taken when Clark visited the University of Arizona in 2018. And he came in and reviewed some of the work that was going on in the laboratory of Dr. Claros Pappas, who's a professor at the university and created the Institute for Cellular Transplantation. Claros is over on the right. And then there's some members of the old team behind me. Steve Neuenfeld, the engineer that brought and set up sonic welding device fabrication at the university. And then Tom Ludavaris, the immunologist who was part of the team, he had been working with Claros already. So at this point, I think that we would really love to get our hands on a cell line that we could put inside these devices. And at the moment, we're making new designs and improved membranes in the laboratory of Claros. So at this point, I want to say to Clark that I felt very fortunate to have met you about 36 years ago. I want to thank you for your help, enthusiasm, and encouragement. And it indeed has been a privilege working with you. Thank you.
So this is going to be a tough act to follow, and I'll try and actually not be repetitive. There is a constant theme that comes out of the presentations, the incredible presentations of today, highlighting the rigor and the impact that Clark had. So I will try to summarize some of that without being repetitive, and the stage has been set up very nicely for me from the previous speakers. Maybe I would say that I did not graduate from MIT. I did all my undergrad and graduate work at Georgia Tech. I got my PhD in chemical engineering under the mentorship of Dr. Thanasis Sambanis, who also did his PhD at the University of Minnesota under Dr. Fredkison, and then a postdoc here at MIT under Dr. Gregory Stephanopoulos. So he had the rigorous training and background both in modeling as well as experimental work. So I was very privileged to learn from him, and then that actually led me ultimately to MIT and the mentorship of Professor Colton, who had a profound impact on my thinking and career and what followed. So I'm very privileged and very honored to speak here in this setting. Just some coordinates, and actually, I would say that I devoted my career in diabetes, maybe unlike some other people who had the zigzag approach or some of the other approaches. So I've been working on type 1 diabetes for about 36 years. I started in 1990 with my PhD, and the work was actually on microencapsulation using engineered cells and oxygen supply, oxygen limitations, oxygen consumption rate, were the theme as well. And I did benefit tremendously by also Dr. Nerem, who was recruited at the time at Georgia Tech to build the first bioengineering program. I did not graduate out of that because there was not a formal degree at the time. So my PhD is in chemical engineering, working on tissue engineering. And Dr. Nerem had invited Professor Colton and also Professor Langer in the lecture series. I had the privilege to actually observe some of the work that they were doing at MIT at the time as a student. How did I get to meet Clark? I was actually at a conference where Dr. Agustiniados, now a doctor at the time, as a PhD student, was presenting his mathematical modeling work with Dr. Colton, and he had a complaint in true fashion, as everybody who works with Clark, very straight approach. Well, the data didn't match the theory. So he had actually theory predicting necrotic course in islets. Let me see if I can use in culture, and his theoretical predictions were not matching the experimental results observed by Hai and Wu and also some of the work that was done by Keith Dion. In other words, the necrotic cores that would have been predicted were much smaller than what were actually observed. So I had the privilege to be working on oxygen consumption rate measurements at the time, and I had scanned the literature, again, talking about the importance of not polluting the literature, but also being very rigorous about that. I realized that many of the measurements that were done on oxygen consumption rate, even though true in terms of the experimental results, may not have been appropriate with respect to the media used. So the long story short is that the experiments were done in nutrient-rich media with amino acids in culture, and the necrotic cores measured actually experimentally were observed in nutrient-rich media, but the oxygen consumption rate measurements used, the reaction rate used in diffusion reaction models, was actually measured in nutrient-free buffers that did not have these amino acids. So it was a lot lower than what is actually in nutrient-rich media. So I did some back-of-the-envelope calculations and figured out that if we use the oxygen consumption rate of islets in nutrient-rich media, the theory would match the experiments and status could graduate. So I decided to reach up, pick up the phone and reach to Professor Colton. You know, I was finishing up a postdoc at the time at Novartis. I did a slightly different detour there, still working on diabetes there. So I picked up the phone and called, and actually Professor Colton picked up the phone. And I said, Dr. Colton, I don't think you know me, but I think I have found a solution to one of your problems, matching the theory with experiments. And I proceeded to describe what I thought was the solution. So he looked, I couldn't see him, obviously. We didn't have Zoom or anything at the time. So he said, you know, you might be right. And I said, well, I think I am. I've done my homework. And he said that actually he was observing and looking at things as I was speaking to him and realized that actually this had some truth to it. So he said, can I offer you a job? I said, well, I have another position, at least offered. I did not accept at University of Pennsylvania. He said, no, you should come to MIT. And of course, I knew of his work and I was very excited about encapsulation. So I said, I would think about it. And then we had to discuss salaries. And I'm not going to mention numbers here, but you know, whatever he was offering at the time of an academic postdoc was a lot less than an industrial postdoc at the time from a Swiss company. So I said, Clark, I really would love to do this, but I cannot survive. I don't have a trust fund, and I really need to work for a living. So he said, okay, let me think about it. So he reached out to Professor Gordon Weir over at Joslin. And between the two of them, came up with something that was workable for me to move to Boston. And the rest, as you say, is history. But that was how I met Clark. And it was actually pretty impressive. I expected that Clark would say, what do you mean we have something that is incorrect or is not, is not accurate? But he actually embraced it. And we worked on this topic for a while. So oxygen consumption rate measurements became very important. Actually, at the time, part of the appointment came from the Islet Transplantation Center at Harvard Medical School. It was a JDRF-funded center with about 40 investigators, mostly from Harvard, and 2 investigators from MIT, Clark and I, which were considered a little odd there. But our job was to actually try and figure out encapsulated cell therapies for the treatment of diabetes at the time, related, with better immunosuppression and immunomodulation strategies potentially coming out from the Harvard groups. So as we were sitting in these meetings, which again, very, very, very impressive group of scientists, Dr. Susan Bonner Weir, Dr. Gordon Weir, part of it, Doug Melton was part of it at the time, just to mention a few. In the meetings, a lot of complaints surfaced about islets. So there's very expensive immunomodulatory protocols, very expensive large animal studies, and nobody knew whether it was, they didn't work initially, so nobody knew, was it the islets, the islet quality, or was it actually our protocols? And several people thought it was the islets, but nobody could put their finger on it. So we came up with the idea of measuring oxygen consumption rate as a rapid way of measuring viable islet volume and predicting whether these islets would work or not. At least we gave people, or have an opportunity to give people islets that had a chance. And then if the immune system would take care of them, that's another story. But at least when they went into the animal, or ultimately the person, they actually worked. So we started with equipment that it was very homemade, with probes that were industrial, because there was no equipment to measure oxygen consumption rate at the time, and eventually miniaturized it, and came up with equipment that actually was sold by a company called Instec Labs. And in true Clark fashion, and also from my training, these chambers were made out of titanium. We found out that if you use stainless steel, it could oxidize and it could consume oxygen instead of the cells and mess up your measurements. The steering bar, instead of Teflon, is glass coated, because if you want to go down to 0 oxygen, Teflon might allow oxygen to diffuse out and mess up your measurements. So this is the kind of level of detail that you don't typically see, or you don't know if you were to purchase this equipment or use it, but that's the level of detail that went into that. We used that equipment and predicted actually how the islets fared, and that became almost a standard in the field. I had to collaborate with many groups in this field, and actually, because of that, Dr. Herring, a very prominent cell therapy person, islet transplanter, along with Dr. Sutherland, recruited me on a faculty position in Minnesota in 2003. And this was a very interesting approach to them. Actually, they were reviewers of the center, and they liked this project so much that they actually voted for further funding, specifically for this, instead of the encapsulation approach, unfortunately. So I moved to Minnesota in 2003, but I continued to work with Dr. Colton and also Dr. Weir and Dr. Susan Bonner-Weir throughout the years. We correlated actually these oxygen consumption rate measurements with transplant outcomes. The FDA really liked that. If you're going for a biological license application, you need to have a viability and potency assay that is predictive of the transplant outcome in this case. And we went along and published several papers on this topic, and ultimately moved it to clinical testing with Dr. Sutherland. Unfortunately, again, Dr. Sutherland is not with us. But he was one of the pioneers in cell therapy in years, and again, working with Clark and Gorton in doing this, we're relating oxygen consumption rates to clinical auto-transplant outcomes, and ultimately with James Shapiro and his team for clinical islet allotransplants. And this led actually to the FDA looking into this for the BLA submissions. I was invited to speak about this potency testing during the BLA filing. Now, the theme of my work, again, is on oxygen delivery and oxygen measurements, and a lot of that is owed to Clark, who actually pioneered this work. And at the time I joined, he was working with the Therasite devices with Haiyan Wu and Stathis Augustiniados and others on delivery of oxygen through implantable oxygen generators. So oxygen delivery is a theme in islets for the reasons Dr. Susan Bonner-Weir shared earlier, and Keith actually shared with his early work. And one of the things we did early on was also to try and figure out a way to simplify islet culture. So if you use the models that Stathis and Clark had created, you would need about 500 flasks to culture an entire islet preparation prior to transplantation. And that was a big problem if you're trying to do a lot of clinical transplants. So we were able to just change that membrane with a silicone rubber gas permeable membrane, very simple solution, and culture an entire human islet isolation in a single vessel as opposed to 500, just by providing the oxygen from the bottom instead of having it diffuse from the top through media. And you could put a lot of media on the top, and you can see here also the tick marks are on the inside in these graphs, and nothing could be published without that. We also proceeded to look at oxygen delivery to organs, and one of the reasons I was excited to move to Minnesota is that, unlike the situation at MIT, in Minnesota, in the Department of Surgery, I was able to get my hands on organs very easily. Dr. Sutherland would be very happy to deliver, whether it was a pancreas, a research pancreas from a human donation for research, or a porcine pancreas for us to study. And we showed that it was impossible really to oxygenate a pancreas from the outside, even at 8 degrees Celsius. So people thought maybe you reduce the temperature, oxygen consumption rate goes down, you don't need oxygen. In fact, the oxygen consumption rate is substantial at 8 degrees, so the penetration depth in there is less than a millimeter. So if you have an inch to go, like in the human pancreas, there is no way to oxygenate the pancreas from the outside. So I have only 1 minute left, it looks like. So I asked Professor Colton if I could continue the work on oxygen delivery and oxygenation. He said yes, that would be great, and we collaborate on that. One of the things that were summarized before is that if you want an encapsulation device to work, it had to be the size of a tabletop in order to provide enough oxygen. And that is something that needs to be addressed, and delivering oxygen locally is a way to have a device that works. A lot of people went to the clinic and failed because they didn't recognize the oxygen limitations and also used the wrong membranes, even though there's publications from Dr. Johnson, from the Baxter days on how to address that. The devices used in the clinic were actually membranes that were known to cause a foreign body response. So not surprisingly, foreign body response is a problem. Oxygen is a problem in vivo. The PO2 is very low. And then ultimately, what I would say is that working with Dr. Johnson and some of the team there, we are moving to the clinic with an approach that I think will help reverse diabetes in patients with a reasonably sized device by delivering oxygen and having optimized membranes for vascularization, but also better mass transfer. I will conclude with the last area that we have worked on recently with Dr. Johnson and Putnam. It's funded by the Gates Foundation, and it's a cell factory approach. And again, a cell factory approach is a trendy work these days. But actually, the work that was done by Baxter and Clark in the 90s was actually a cell factory approach. But in this case, we have human ARP19 cells produce an antibody against HIV, and you have shown that you can have a production rate that is dependent on the size of the device, and actually you can hit target product profiles with a 3 square centimeter device, size of a quarter, with enough production of antibody to the human. So that is a pretty important finding, and this actually devices vascularize very well. You can see the endothelial cells lining blood vessels at the surface, so these devices do not fibrose, and this could be translated, I believe, to the clinic soon. And I would like to conclude this presentation by thanking Professor Colton for his mentorship, his work in the field, the mentorship of everybody else, and the tremendous impact on real world health issues that would treat people in generations to come. Diabetes is a tough nut to crack, so we're not there yet, but it will be there soon. So for Dr. Susan Bonner, unfortunately, Dr. Gordon will not be with us to see this, but I believe we will address this problem and have a solution on cell-based therapy with encapsulation in the very near future. With that, thank you for your attention, and thank you, Clark, for all you've done.